Thanks so much for inviting me to speak at the conference this year, and thanks to all the organizers for putting it together. I'm really excited to tell all of you a little bit about my work, which is trying to understand learning algorithms in cortex cerebellum circuits. So to jump into a bit of background, I think that while it's well appreciated that the neocortex has expanded dramatically over the course of mammalian brain evolution, it's a lot less well appreciated that the cerebellum has really kept pace with that expansion, so that consistently across species, you find about 4 times as many neurons in the cerebellum as there are in the cortex. And this sort of tandem enlargement of these two brain areas has come kind of at the expense of the rest of the brain, so that in humans, cortex and cerebellum alone make up about 99% of all neurons. So there's this close evolutionary link, and these two structures are also really closely linked neuroanatomically by a universal set of connections. So throughout the entire neocortex, not just motor, but auditory, visual, cognitive, all parts of the cortex, all subcortically projecting layer 5 pyramidal neurons send a collateral to the basal pontine nuclei, and all of those cells project up to the cerebellum, where they contact granule cells, which alone make up more than half the neurons in the brain. And there's an analogous pathway back from the cerebellum to the cortex via the thalamus. And so to me, the universality of these networks, universal across the cortex, universal across mammals, suggests that there may be some kind of basic and conserved algorithm implemented by these circuits that underlies sort of the deep evolutionary conservation. And I think the prevailing model of how these networks might work is inspired largely by the motor domains of these circuits, with the basic idea being that the neocortex, in order to control body movement, actually needs predictions of the future outcome of our actions, and that's because of the long latencies that separate our brains from the body periphery, so that our sensory information is always old. And various lines of evidence suggest that the cerebellum may be providing this kind of prediction or simulation function. And I think it's easy to imagine that that could be generally useful even in contexts that have nothing to do with body movement. And so as a first step towards trying to understand this big high-level computational model of cortex cerebellum circuitry, I've been focused on trying to investigate this cortex to cerebellum pathway and how it evolves during novel skill acquisition. Now, the prevailing model for the function of this pathway was established largely decades ago by Marr and Albus, with the basic idea being that mossy fiber inputs to the cerebellum, which mostly come from the neocortex, undergo a massive numerical expansion, about 1,000-fold, out into the cerebellar granule cell layer, but the connectivity there is really sparse. So each granule cell only gets about 4 synaptic inputs, which you can contrast to a typical neocortical neuron, which might get on the order of thousands of inputs. And those neurons then converge down into Purkinje cells. So the basic idea is that representations that mix different types of information up in the cortex could then be separated out in the higher dimensional granule cell space. So for example, imagine you have 2 different neocortical neurons on the X and Y axes of this plot, and you representing 2 different stimuli shown in red and blue, and you can't really tell these 2 stimuli apart in that representational space. Perhaps if you had the same information represented in the higher dimensional granule cell space, you could tease those 2 things apart. So this is sort of a classical machine learning perspective on this really basic and conserved biological circuit. Now, when I got started on this work, most of what was known about the anatomy of this pathway came from species like primates, monkeys, rats, a bit in cats, but very little in mice, and also very little using some of the more modern, strictly disynaptic tracing techniques. And so I wanted to leverage some of these techniques to investigate this pathway in mice, which are my model system. And this is basically a 3-virus, 3-injection strategy, the upshot of which is that we can label all of the neurons throughout the cortex that disynaptically contact a particular region of the cerebellum via the pons. And basically what we found is that the entire cortex sends these projections. So prefrontal areas like the orbitofrontal cortex, more classical somatosensory and motor regions, limbic areas of the cortex like the cingulate, association areas like retrosplenial and parietal cortex, and even primary sensory areas like visual and auditory cortex. So this really does seem to be a universal disynaptic projection from the entire neocortex to the cerebellum in mice, which I think makes mice a really great model system for trying to understand this universally conserved mammalian pathway. And I'm going to tell you a little bit about several of my attempts to understand this pathway at a functional level, starting with the development of some four-limb skill learning tasks for mice that require these circuits, then moving on to a study of cerebellar granule cell physiology during the execution of these behaviors. Then I'll describe a dual site simultaneous two-photon imaging strategy to monitor cortex to cerebellum transmission and how it evolves with learning. And then I'll switch gears a little bit and tell you about some of my work investigating synchronization in the climbing fiber system and how that might fit into broader cortex cerebellum dynamics. So starting with the behavior, really a lot of what's been learned in motor neuroscience has come from these skilled arm reaching tasks where subjects grasp the handle of a robotic arm and have to direct it in various ways while the experimenter can push and pull on the subject and see how they respond and ultimately learn to compensate. And I think some of the key virtues of these tasks that have made them so influential is that they can be kinematically simple, the movement's highly stereotyped, but we can arbitrarily perturb or constrain the subject's motion in real time using the robotic controllers that drive these devices. And this allows us to devise a really wide variety of assays for different components of behavior and learning. So after working with these behaviors as an undergrad at Harvard with Maurice Smith, when I came out to Stanford, I really wanted to adapt these behaviors for mice, where we'd also have access to the tremendous toolkit for brain circuit interrogation in vivo. So we developed the first adaptation of these devices that have been used for decades in primates for mice, while retaining this key feature of closed loop real-time force control to arbitrarily perturb or constrain the arm motion of mice in real time. So you can see what this looks like in a behaving animal. This is a head-fixed mouse that uses its right forelimb to grasp the handle of this device. And on different trials in this particular task, you'll see the animal push in different directions in order to get a delayed water reward. And animals will execute hundreds of these trials in the session. And at the same time, we can monitor or perturb brain activity in one or multiple areas and do so repeatedly over weeks or days of learning. Now, all the tasks that I've looked at have a very simple general structure where the animal executes a movement, and then after a delay, if it's successful, it gets a water reward, and after another delay, the robot automatically returns the handle to the animal so that it can self-initiate the next trial. So the simplest version of this task is just a basic operant task where the animal pushes forward to get a water reward. A more complicated version has the animal push forward and to the left or forward and to the right, depending on trial type, as a way to study how the brain learns to generate complex sequences of movements. You can study unconstrained movements where the animals learn to aim towards a particular rewarded target zone. And of course, you can imagine training animals to aim towards multiple distinct targets as well. And all of these behaviors take on the order of several weeks for animals to learn, which is a great timescale for using in vivo brain imaging to monitor the entire time course of learning. And I'll just mention that it's very easy to extend these behaviors to bring in more cognitive decision-making components, in this case in animals viewing an LED screen, and has to associate different visual patterns with pushing in different directions. So we started just by showing that these behaviors actually require these cortex cerebellum circuits using double transgenic mice that express channel rhodopsin throughout all the inhibitory neurons in the brain, and then exposing cranial windows over both the premotor cortex and the cerebellar cortex to alternately disrupt activity in one brain area or the other during behavior. So what you see is that in the first minute, the animal gets about 10 rewards. And then when I start disrupting activity in the premotor cortex, the success rate falls close to 0. Similarly, if we disrupt activity in the cerebellar cortex, success also falls close to 0. So we do think that both of these brain areas are critically important to the execution of these four-limb skill learning tasks, which I think makes them a great set of model behaviors to now dive into the physiology. And as I mentioned, we started by looking at cerebellar granule cells, neurons that make up more than half the cells in the brain, and hadn't really been looked at in the context of motivated behavior. So we didn't have answers to some very basic questions about what these neurons represent and how they represent it, due to longstanding technical limitations related to how tiny and densely packed these neurons are. So you can't really use classical extracellular electrophysiological techniques to reliably isolate neighboring neurons from one another. So instead, we developed a transgenic two-photon imaging strategy using triple transgenic mice that express the green genetically encoded calcium indicator GCAMP6 throughout all granule cells of the cerebellum, but not other cell types, for example, not the Purkinje cells shown in red, and then use two-photon imaging to optically section down into this granule cell layer. And you can see that for the first time, we were able to visualize hundreds of these cells simultaneously and to monitor them repeatedly over days or weeks. And you can see from the scale bar here just how critical it is to have the high optical resolution of two-photon microscopy to be able to reliably disambiguate neighboring neurons from one another. So although we can visualize hundreds of these cells at the same time, typically only a subset of those have detectable fluorescence changes. And so we use a non-negative matrix factorization algorithm to identify the spatial locations of those active cells. For any one of those neurons, we can extract its time-varying fluorescence trace, here shown in z-scored units, cross-reference that with the times of the individual behavioral events of interest. In this case, I'm showing you the forelimb movements, align all of those different movements, and for example, produce what we call the trial averaged activity pattern for this neuron, which tells us something about when this cell tends to activate during the execution of this task. And in particular, I focused on this simple push for reward operand task that I described to you previously as a way to have sort of a nice mix of classical sensory motor variables and cognitive variables related to reward delivery and the general temporal sequencing of this task. So here I'm showing you the animal's hand position over time. You see that they start from position 0, push about 8 millimeters, and then after a 1-second delay at the time of this dashed vertical line, they get a water reward. We also monitor the animals licking, which is shown in black. You see that these animals start licking robustly during the delay period when they're anticipating this upcoming reward, and they continue licking to consume the reward. So here's what we found in the granule cell layer. These are all of the neurons that we recorded from a single mouse. Each row shows the time-varying fluorescence of one granule cell aligned to movement and color-coded by the magnitude of activity on the right. And then I've sorted the different cells by the time of their peak activity. So for example, these neurons have peak activity prior to movement. Then there's a whole bunch of cells whose activity tightly time locks to the forelimb movement itself. So these might be sort of classical movement-related cells. But beyond that, we found many neurons that didn't become active until the delay period when the animal was waiting for this upcoming reward, or even until after the reward was delivered, which we thought was kind of interesting because we hadn't seen this type of signaling reported before in the cerebellar literature, and we wanted to dig a little more deeply into what these neurons might be representing. So on a random subset of trials, I unexpectedly withheld the reward from the animal. And here I'm showing you for a single example granule cell, its fluorescence on the y-axis as a function of time on the x-axis aligned to reward delivery here at time 0, and broken down by trials where we delivered the reward in blue or unexpectedly withheld it in red. And you see that this neuron has a larger response when the reward's delivered than when it's withheld. Now, conversely, we also found granule cells that selectively activated following the omission of an expected reward, which we thought was interesting because there's no external sensory event to trigger this type of response. And for me, the most interesting types of neurons that we identified, we called reward anticipation cells. And basically what we showed is that these neurons turn on during the delay period when the animal begins anticipating this upcoming reward. At time 0 in blue, these neurons turn off if you deliver the reward because anticipation is over by definition. On the other hand, if you withhold the reward in red, these neurons stay on longer, as though the animal's hoping it might still get rewarded until eventually it gives up and these neurons turn off. Now, to really show that these responses were general and not specific to this particular forelimb task, we also looked at a very different task, a basic Pavlovian task where you play a tone that predicts a reward at a fixed latency, and found a very similar panel of granule cell activity profiles, including these reward anticipation cells that I just described. Now, to really nail down that these neurons represent anticipation and not something about, say, the sensory or motor experience of receiving a reward, we also delivered unexpected rewards that were not preceded by a tone and therefore could not be anticipated, reasoning that an anticipation cell should therefore stay silent. And that's what we found, that these neurons have no response to unexpected reward, despite the identical sensory motor experience, the same water stimulus, rewarding outcome, licking and swallowing motor responses, which we think is really strong evidence for these neurons representing this internal cognitive state of waiting for an upcoming reward. It was really the first evidence for this type of signaling in the cerebellum. And I'll just briefly mention that in a more recent study, we also found that some granule cells have general responses across both a air puff to the face and an electrical shock to the tail, despite the very different sensory nature of these two experiences, which suggests there might also be granule cells with generalized aversion responses. So to briefly summarize this first section, we found that granule cells represent a broad diversity of both classical sensory motor and cognitive and prominently reward expectation related signals. And we think that this is a reflection of the broad diversity of neocortical areas that all converge in these regions of the cerebellum. And even though this was the first evidence for this type of signaling in the cerebellum, in the years since this study, a number of other labs have found similar types of reward expectation encoding in other parts of the cerebellar circuit, including the climbing fiber system and cerebellar output, and shown how cerebellar output might impact downstream brain reward processing centers. So with this basic understanding of granule cell signaling, we now wanted to investigate how this related to the upstream neocortical activity. And so building on the previous transgenic two-photon granule cell imaging strategy, we used quadruple transgenic mice that also expressed GChem throughout layer 5 of the neocortex, in addition to granule cells of the cerebellum, and then devised a surgical strategy to expose both of these populations in single animals, and adapted a dual-site two-photon microscope to have full translational and rotational degrees of freedom that are needed to simultaneously access both of these brain regions reliably and repeatably in individual animals. And you can see that for the first time, we're able to simultaneously record cortical output neurons and cerebellar input neurons, which we did in the context of this four-limb movement sequence planning task that I described before. And putting all that together, you can see what one of these experiments looks like in a behaving animal. On the right are cerebellar granule cells, similar to the movie that I showed you in the previous study. And on the left are premotor cortical layer 5 neurons. And you're actually viewing these cells through an implanted optical microprism, which is why you see their apical dendrites rising up towards the peel surface. Typically in one of these movies, we have access to about 80 active cells of each cell type that we can monitor simultaneously and repeatedly over weeks of the learning of this task. So I'll start just by telling you a bit about what we saw upstream in the cortex. So these are two individual premotor cortical layer 5 neurons with their fluorescence on the y-axis as a function of time on the x-axis, aligned to the turning movement that I just described, and broken down by left trials in red and right trials in blue. So for this first cell, you see it turns on about a second prior to movement, selectively before left, but not right trials. Conversely, this neuron turns on about a second prior to right, but not left trials. So these are sort of direction selective movement planning related cells. Now, for other layer 5 neurons, when we align their activity to reward delivery at time 0, we found that they tightly time-locked to or even slightly anticipated reward, in this case selectively following left trials, or for this neuron selectively following right trials. So these are sort of trial-type selective reward-locked cells. Now, we were really surprised that when we looked downstream in the cerebellum, despite the very different nature of the cerebellar circuit, individual granule cells recapitulated very similar single-cell activity profiles, including these direction selective movement planning related neurons and these trial type selective reward locked cells. And in fact, quantitatively, these different types of selectivity were just as prevalent and robust downstream in the cerebellum as they had been upstream in the cortex. Now, because these are simultaneous recordings, I really wanted to dig into the moment-by-moment covariations in cortex cerebellum activity. And I think probably the most surprising finding to emerge from this study is that even though we had access to only a relatively small number of neurons in these two brain areas, there were pretty good odds of finding individual layer 5 granule cell pairs whose moment-by-moment activity fluctuations strongly covaried. And in fact, we went on to show using optogenetic inhibition of the basal pontine nuclei that these correlations are contributed at least in part by transmission through this cortex cerebellum pathway. Now, having said that, we thought that the odds of finding these highly correlated pairs were a bit too high to be sort of a priori hardwired pre-existing state of the circuit. Maybe instead, this is telling us something about the evolution of cortex cerebellum transmission during the learning of this novel skill. Now, historically, there hasn't been a way to address this type of question in neuroscience to track the coevolution of activity in two distant brain areas in fixed sets of neurons repeatedly over weeks of learning of a novel task. But using this dual site two-photon calcium imaging strategy, we're able to track a fixed ensemble of neurons in both the cortex and the cerebellum simultaneously and repeatedly over weeks. And what we found is when you have one of these highly correlated layer 5 granule cell pairs, if you track this specific pair of neurons a few weeks back in time, those correlations were generally absent. And this was true overall, that over the course of learning, it just became increasingly common to find these highly correlated layer 5 granule cell pairs. Now, at the same time that these correlations were emerging, we also found that these two brain areas started to develop a similar set of task encoding activity patterns. So I'll give you some examples of what I mean by that. Here's an individual cortical layer 5 neuron at the end of learning, robustly responds to left but not right turn movements. And if I track this specific cell a few weeks back in time, that selectivity was absent. Now, analogously, here's an individual cerebellar granule cell that robustly anticipates reward delivery following right but not left turn trials at the end of learning. And a few weeks earlier, it had basically no task-locked activity at all. And both of these things were general trends that over the course of learning, more and more neurons in both brain areas became both temporally locked to particular phases of the task and also selective for trial type. So the broad picture that we came away with from this study is that early on, there's a relatively diverse set of activity patterns in both the cortex and the cerebellum with relatively little overlap. And over the course of learning, these two brain areas are actually driven down onto a narrower set of activity patterns that's more shared between the two brain areas. And that largely reflects the emergence of these common task encoding activity patterns, which is really our first glimpse at the cellular level of the coevolution of activity in the cortex and the cerebellum during the learning of a novel task. So I'm going to switch gears a little bit and tell you about some of my investigations of the climbing fiber system and how synchronization in that system might more broadly fit into cortex cerebellum circuitry. So I've been speaking about this layer 5 to granule cell pathway, but the other major input pathway to the cerebellum is the climbing fiber pathway that arises in the inferior olive and also gets indirect neocortical input. And in classical theory, the distinction between these pathways is critical to cerebellar function because a Purkinje cell gets about 100,000 granule cell inputs, but just a single climbing fiber input, which is thought to instruct the Purkinje cell as to which of all of these granule cell inputs is most informative in different behavioral contexts. And so it becomes really key to understand the interplay among these different information sources. So we recently wrapped up a two-photon mesoscope imaging study where we recorded the activity of hundreds of cerebellar climbing fibers simultaneously across a 4 squared field of view repeatedly over days as animals acquired this four-limbed aiming task that I described at the beginning of the talk. And one of the most salient features that emerges during the learning of this task are these big synchronized complex spiking events, as well as these big synchronized silences. And we've become really interested to try to understand how a large network of neurons like this can undergo a rapid synchronization transition. And there are two really key things to know about the olivary circuit for thinking about this problem. The first is that individual olivary neurons have ongoing subthreshold membrane potential oscillations of around 10 Hertz that are sort of autonomous between neurons. But different olivary neurons are electrically coupled to one another by an extensive system of gap junctions. And in fact, it's thought that the cerebellum may have the ability to modulate the strength of that gap junctional coupling. Now, what we recently realized is this general network architecture is highly reminiscent of a very influential and also very well understood model from applied math and physics called the Kuramoto model of coupled oscillators. Basically, you have a system of autonomous oscillators that can be variably coupled to one another, so that at low coupling, the oscillations are basically incoherent, and as the coupling increases, they converge to a common frequency and phase. Now, one of the key things that's known about this system is that in its dynamics, there's actually a critical value of the coupling, below which you're in basically an asynchronized regime and above which you rapidly converge to full synchronization. So sort of a state transition, a discrete transition in dynamics. And we were interested to understand how a network of spiking neurons simulated on top of this classical coupled oscillator model might behave in a few different relevant regimes. In particular, how it might respond either to a burst of shared input, which I'll show in blue, or to a transient rise in the network coupling, which I'll show in red. So with a burst of shared input, of course, you see many more spikes, but they're not actually any more synchronized. And that's because the underlying oscillations remain incoherent. Now, if you also have a transient rise in coupling, now you start to see these large synchronized spiking events, but also critically, these big synchronized silences, which together really beautifully recapitulate some of the key findings in our in vivo two-photon mesoscope imaging data in expert mice performing our four-limb aiming task. And since then, we've now started to move on to try to understand how these climbing fiber synchronization transitions fit into the broader cortex cerebellum circuitry and dynamics that I've been speaking about earlier in the talk. So to briefly summarize what I spoke about today, we found that granule cells transmit a broad diversity of both classical sensory motor and cognitive and reward-related signals. And then by developing a dual-site two-photon imaging strategy, we showed that these granule cell signals seem to recapitulate the full range of premotor cortical behavioral information, but that furthermore, these two brain areas actually converge to these common behavioral representations over weeks of novel skill acquisition. And in parallel work, we've shown how the climbing fiber system develops these big synchronization transitions during learning. And we've now started to work to try to understand how this fits into broader cortex cerebellum dynamics. Now, more broadly, my hope is that some of this work will help us to start to sort of shift the paradigm towards thinking of cortex and cerebellum as more of a joint dynamical system, and to try to understand how these two very different circuits conspire to learn these novel cooperative neural dynamic activity patterns that we think may underlie the acquisition of novel skills. And with that, I'd like to thank my postdoctoral advisor, Li-Chun Lo, my optical engineering collaborator, Tony Kim, and his advisor and my graduate advisor, Mark Schnitzer, Jonathan Cadman and Surya Ganguly for several theory-related collaborations, including the Koromoto oscillator model, and Boxian Zhao for some work on cerebellar connectomics that I didn't have time to speak about today, as well as my funding sources. And I'd also just like to make a brief plug to mention that I'll be starting a lab at the NIH in September, and I would love to have any talented scientists interested in working on cortex cerebellum circuitry contact me. Thanks very much.